Hello and welcome to week 3 for plant biology at NICC. And this week we're going to be looking at chapter 3 in the Concepts of Biology textbook, and we're going to be looking at cells. Cells are the fundamentally smallest unit of life. We can have life that's definitely alive but is only made up of one cell, or we can have life that is multicellular, made up of many different cells. But anything smaller than a single cell, and biology does not consider it alive. When we're looking at these very small things, we need specific tools for the trade, and those tools are known collectively as microscopes. There's a number of different types of microscopes. Dissecting microscopes are used for looking at, say, larger sorts of objects. If you want to look closely at a leaf or an insect or something like that, we use a dissecting scope or a stereoscope for that job. When we're looking at some very small things like cells, we use what are called compound light microscopes. And these are probably the sorts of microscopes that you may or may not be familiar with. But when you think about a microscope, this is probably what mental image you pull up in your mind. Usually they have one or two ocular lenses that you look through. You have a little glass slip, and light gets sent through a sample on this glass slide, and you see a magnified image of it. These compound light microscopes are almost ubiquitous in college classrooms. Any sort of medical or veterinary lab is probably going to have one or many compound light microscopes in it, and they're a great tool for using to visualize what cells look like in real life. So we can look at the interior structures of cells, the exterior structures of cells using compound light microscopes. If we want to look in much more detail and much higher magnification, we can use what are called electron microscopes. And there are two main different types. We can have transmission electron microscopes, which send a beam of electrons through a very, very thin sample and pick up an image that way. Or we can have what's called a scanning electron micrograph, which scans the outside surface of a very small object, and you can get a very high resolution three-dimensional image from the scanning electron microscope. Both of them are very valid tools for the study of cells. Or actually, all three of those are very valid. The compound light, the scanning, and the transmission electron microscopes when we're looking at cells. Most commonly, though, especially in a college classroom, we're going to be using compound light microscopes to view cells. We're not going to actually use the scopes in this class since it's online, but those resources are definitely available. Being able to see these very small objects really allowed the development of what's known as the cell theory. And the cell theory states that all cells come from other cells, and all living things are composed of cells. And that's a pretty basic sort of understanding of how life exists on this planet, but it's really fundamental in understanding how life exists. This cell theory dates back quite a long time, but microscopy was really has been known for quite a long time as a way of studying very small things. Robert Hooke was a experimental scientist who first coined the term cell when looking at these very small structures of organisms, by looking at a very thin section of a piece of cork that he'd cut through an early microscope. So these early microscopic experiments led to the development of our understanding of life as being composed of these very small units that we call cells, and that all life is derived from previous cells. When we're looking at different types of cells, we can break out cell types in two main categories. We can have what are called prokaryotic cells, which are more primitive, or we have eukaryotic cells, which are more recent. The prokaryotic cells are things like bacteria and the archaea. The main thing that separates the prokaryotes from the eukaryotes is that prokaryotic cells do not have membrane-bound organelles. We'll talk more about organelles a little later on in this chapter. Prokaryotic cells also tend to be very much smaller than eukaryotic cells. If you think of bacteria, bacteria cells are very small compared to something like a single-celled organism like a paramecium, which is a relatively large single-celled organism. Also, if you think about cell size, A chicken egg is a single cell. Sort of a non-traditional way of thinking about a cell, but it is truly a single cell. Frog eggs are a single cell. Human eggs are a single cell, although we generally can't see those without a microscope. But most of the cells that we see tend to be too small to be seen with the naked eye, and so we use these microscopes as tools to see them. In 3.3, we start talking about eukaryotic cells, and for most of the purposes of this class, we're going to be more concerned with eukaryotic cells since plants are among the eukaryotes. In the visual connection area of the textbook, they have a really nice diagram of two different cells, and these are just generic sorts of cells. One is an animal cell on the top, and the other is a plant cell. So for this course, let's focus on the plant cell at the bottom. And this image really does a good job of showing and mapping out the basic fundamental parts that you can see within a eukaryotic plant cell. Let's start on the outside of that image. On the outside of that image, in this case anyways, is a green sort of shroud. And that green shroud is known as the cell wall. The cell wall is outside of the bounds of the cell because what's directly inside the cell wall is the plasma membrane. The plasma membrane is what separates the inside from the outside of the cell, but the cell wall is a structure built by the cell to provide structure to the cell. In the case of plants, the cell wall is made up primarily of cellulose. That provides a rigid framework that the plant cell exists within and provides protection for the cell as well as structure. Just inside the cell wall, again, we have the plasma membrane, and I think I'm going to do a mini lecture on the plasma membrane specifically so we don't soak up a bunch of time here with the plasma membrane, but the plasma membrane is, again, what separates the inside from the outside of the cell, and it really becomes the gatekeeper for what goes in and what leaves the cell. The plasma membrane is critically important for cells. I think I'm going to spend a little time elsewhere on that. Within the plasma membrane, we have what's known as the cytoplasm. And the cytoplasm is sort of the goo that is within the cell. Within the cytoplasm, we also have organelles. And the primary sort of brain of the cell is the nucleus. The nucleus is where the DNA is housed and where the information that the cell requires to do all the cell things it needs to do to survive takes place. Other organelles within the interior of the cell, for example, are endoplasmic reticulum. Endoplasmic reticulum is involved in the production and processing primarily of proteins and other biomolecules. We have Golgi apparatus, which are involved in the transport of these molecules. In plants, we have chloroplasts. Chloroplasts are organelles that are the site of photosynthesis. So the ability of plants to take sunlight energy and convert it into sugars occurs at the site of the chloroplasts. Utilizing that energy that the cells make in the chloroplasts takes place in the mitochondria. Mitochondria are known as the powerhouses of the cell, and they're involved in breaking down those sugars and providing energy for the cell. So chloroplasts and mitochondria are both critical components in the organelle structure of these cells. Also, a major component of plant cells is what's known as the central vacuole. And this is really just basically like a fancy water balloon inside the cell. This vacuole fills and swells with water, and that puts pressure on the plasma membrane against the cell wall, and it provides what's known as turgor pressure. If you think about a plant leaf, it's flat. Here, I'll get my hand. As it wilts, as it runs out of water, that plant leaf starts to droop, right? It's losing turgor pressure. If you water that plant and it hasn't died and then it absorbs, as the plant absorbs that water and fills that central vacuole with water, it exerts pressure on the cell membrane against the cell wall, and the leaf starts to re-emerge or build itself back out in this sort of normal sort of structure. And that turgor pressure is important for plants to be able to stand upright and compete with one another for sunlight resources. There's a number of other smaller organelles, ribosomes and things like that, that are are important, but you can certainly read about those, and if you have any questions, please post on the discussion board and ask those questions about them. Another important component of cells is the connections between cells. Obviously, our cells are all connected just like the cells of plants are, and these intercellular junctions are how cells communicate with one another across the span of a large organism very often. And so sending chemical signals requires these connections between cells. In the case of a multicellular organism, the single cell does not exist very effectively outside of the broader organism itself. All right, that sort of covers very briefly the different parts of cells and kind of what we know about cells. Again, I think I'm going to post another video today specifically on the plasma membrane and moving things through and across the plasma membrane, but that's I think it's a little more in-depth of a concept that can benefit from a more focused recording on my part. So again, go through chapter 3 of Concepts of Biology and learn what you can about cells.